Good morning, everyone. Let's talk about what's going to go on this week. Firstly, as usual, there's no scheduled formal Zoom meeting, but if you need me, please reach out and we will set one up in some afternoon office hours. And if you have any questions about anything at any time, email me. But let's go through the week here. Monday, you should take the time to watch the Crossing Over Edpuzzle video, work through that. Tuesday, there's an Independent Assortment Edpuzzle video. Wednesday, there is a vocabulary assignment where you take the important terms from chapter 10 having to do with meiosis and other genetic things that happen during meiosis. You will take each word, give a definition on the top box, use it in a sentence on the bottom box, and do a Google search to find an image that relates to that word and paste it into the box on the right. And then Friday, there's a video you need to watch called The Greatest Discoveries in Genetics. You'll need to open up video on one side of your screen and open up the questions Google Doc on one side of the screen and answer the questions as you go about these people that have had an important impact in the scientific world of genetics. It's kind of a fun watch to learn these things, but yeah, get those things turned in and you will be good to go. But yeah, let's go ahead and take a second here and look at today's lesson. We're talking all about Mendelian genetics, gene linkage, how things kind of work together, and then what does polyploidy mean? So let's take a second and look at those things. All right, we are going to talk a little bit about Gregor Mendel, who is considered the father of genetics. Let's begin. So the passing of traits to the next generation is called inheritance or heredity. And Gregor Mendel published his findings on the method of inheritance in garden pea plants. So he took these pea plants and started noticing that based on how they were kind of bred together, they looked different. And he started thinking, why? Why do they look different? What's going on here? Well, cross-pollinated pea plants, which normally self-fertilize, he noticed that they rigorously followed various traits in the pea plants that he bred. So by studying this, he began the study of genetics, which again is the science of heredity. One trait that Mendel noticed was seed color. Some plants always produced green seeds, others always produced yellow seeds. And he crossbred the green and yellow seed plants, and then he called the green seed and yellow seed plants the parent generation or the pea generation. So the ones that you begin with are the parent generation or pea generation. As they were crossbred, they had their offspring, so to speak, and the offspring had offspring. These are the F1 and F2 generations. So the offspring of the pea cross-pollinated plants were the initial or first filial generation, or F1. The second filial generation, or F2, is the offspring of the F1. And in Mendel's peas, the green seed trait disappeared in the F1 generation, but reappeared in the F2 generation. So the F2 generation showed a ratio of 3:1 yellow to green seeds. So he noticed the first time the yellow and green bred together, only yellow came out of it. But then when he crossbred the yellow ones from that second generation, yellow and then some green appeared again. So he started to think, what happened? He only crossed yellow together, so why are there green? And that's what led to the next big question for him. So the F1 and F2 generations, during that, Mendel studied 7 different traits: seed or pea color, flower color, seed pod color, seed shape or texture, seed pod shape, stem length, flower position. And in all these cases, Mendel found that the F2 generations showed a 3:1 ratio of traits. No matter what, there was always a 3:1 ratio in that second offspring, which is the third generation, or the F2 generation. So Mendel concluded that there must be 2 forms of the seed trait in the pea plant and that each was controlled by a factor. So the word allele, you're going to hear that a lot from now on. An allele is an alternative form of a single gene. So the gene for yellow seeds and the gene for green seeds are different alleles for the same gene. Now, alleles can either be what's called dominant or recessive. Dominant alleles control the traits that do appear in the F1 generation. that they're dominant, they're going to appear. Recessive alleles were masked in the F1 generation. And based on how the DNA came together is what determined whether they were going to express the dominant or recessive traits. And we'll see a little bit about that in a second. So dominance, when modeling inheritance, the dominant allele is represented by a capital letter. We're going to use Y here. And a recessive allele is represented with a lowercase letter, and in this case, a lowercase y. An organism with two of the same alleles for a particular trait is called homozygous for that trait. So either two big Ys or two little y's. If the traits are the same allele, it's homozygous. An organism with two different alleles for that trait is heterozygous. So big Y, little y, they are different alleles for the same trait. In heterozygous individuals, the dominant trait is going to be observed. So obviously in homozygous, if you have two capitals for homozygous, it's going to be dominant. If you have two lower cases, it's going to be recessive. And if you have a capital and a lowercase, the dominant takes over and will be observed. So heterozygous is always showing the dominant trait. And then we have the words genotype and phenotype. So the appearance of an organism does not always indicate which pair of alleles it possesses. And an organism's allele pairs are called the genotype. So genotype are the genes, like what pair of genes do they have? Now the observable characteristics or the outward expression of the allele pair is the phenotype. So when we go back to those seed pea plants, they used Y for yellow. So big Y would be the genotype, and yellow would be the phenotype. The observable trait is the phenotype. So the genotype is the genes. So big Y. Phenotype is what you see. So yellow. Now, Mendel's law of segregation states that the two alleles for each trait separate during meiosis. So if you remember during meiosis, we saw those chromosomes pull apart. The two alleles are separated. During fertilization, two alleles for that trait unite. Heterozygous organisms are called hybrids. So when those things come together, we call that a hybrid. Now, when we start to cross the genetics in this, and we're looking at it in terms of these pea plants here, a monohybrid cross is a cross that involves hybrids for a single trait. And a dihybrid cross is the simultaneous inheritance of more than one trait in the same plant. So hybrid crosses, we're just talking about one specific trait. Dihybrid crosses, we're talking about multiple traits at the same time. Now, we also have what's called the law of independent assortment. This law states that there's a random distribution of alleles occurring during gamete formation. So when your gametes are being formed, there's kind of no rhyme or reason as to why the genes are distributed the way they are. There's so many possible combinations that it's completely random and you can't really pinpoint what's going to happen, which ones are going to be given. Genes on separate chromosomes sort independently during meiosis, and each allele combination is equally likely to occur. Like, it's just as probable to get any of these as it is the other. Now, Punnett squares are what we can use to predict possible offsprings of a cross between two genotypes. So if we have like the ability to roll your tongue, some people can do that. And when I say roll your tongue, I mean do this. Some people can do that. Some people cannot do that. That's actually a genetic trait. So we're going to do capital T for the ability to be able to do that, and lowercase t for the inability to do that. And we're going to cross two heterozygous individuals on the top of the box and on the left side of the box. And basically you cross multiply and put the results inside the box. So big T times big T, you get two big T's. Big T times little t, you get big T, little t. Big T times little t, you get big T, little t. And little t, you get little t, little t. So you can see here, homozygous dominant is always dominant. Heterozygous is always dominant. And then homozygous recessive is the only recessive. So that's why it's 3-to-1 no matter what, which we said earlier. So the number of squares is determined by the number of different types of alleles produced by each parent. Typically, we're just going to have 4 squares here. Now, in a dihybrid cross, things get a little more complicated. You don't have to worry too much about this. I'm not going to make you do any of these. Next week, you probably will do a regular 4-squared Punnett square. But for now, this dihybrid cross, I'm just going to explain what it is. So we have 4 types of alleles from the male gametes and 4 types of alleles from the female gametes that are produced. And if you look through it, it produces a ratio of 9 to 3 to 3 to 1. So 9 of one kind, 3 of one, 3 of one, and 1. Now, probability, when we're talking about probability, the inheritance of genes can be compared to the probability of flipping a coin. Actual data might not perfectly match predicted ratios. If you flipped a coin 50 times, you might get heads every time.
The chili cook-off will begin in 8 minutes, and she asks, please send your email. Thank you.
If you flip a coin 50 times, you could get heads 50 times, or you can get tails 50 times, or you could get heads 10 times and tails 40 times. Like, that's actually how it works with genetics. It's random, but we are able to take mathematical ratios to figure out the probability, like what might be extremely possible to happen. And Mendel's results were not exactly a 9 to 3 to 3 to 1 ratio, but the larger the number of the offspring involved, the more likely it will match the results predicted by a Punnett square. That's why using something like pea plants is good, because you get a whole bunch of them. With humans, it's not exactly the same, or animals, because typically you get 1 offspring. With animals, you might get more than 1, but it would take a lot longer and a lot more time and effort to study that in a human. That's why something like this is wonderful to study with something like a pea plant. Now, genetic recombination, the new combination of genes produced by crossing over and independent assortment is called genetic recombination. Combination of genes due to independent assortment can be calculated using the formula 2 to the nth power, where n is the number of chromosome pairs. So any possible of male gamete can fertilize any possible of female gametes. So the possible combinations after fertilization are 2 to the nth power times 2 to the nth power. So humans have 23 pairs of chromosomes, so 46 chromosomes. A male gamete gives 23 and a female gamete gives 23. So in humans, that would be 2 times the 23rd power, or excuse me, 2 to the 23rd power times 2 to the 23rd power to get you the all possible combinations after fertilization in a human. It's a very large number. Now, gene linkage, genes located close to each other on the same chromosome are said to be linked. They usually travel together during gamete formation, and gene linkage results in an exception to Mendel's law of independent assortment. If you look through these pictures here, they've kind of used color to indicate where similar genes are. And you can see that the colors are lined up in each of these. So as they travel, those similarly linked genes are going to travel the same way. When centromeres separate and gametes form, the same type of genes are going to go together so that later they can be lined up again similarly. We also have what's called chromosome maps. Crossing over occurs more frequently between genes that are farther apart. Crossover data can be used to create chromosome maps, which are depictions of how genes are arranged on a chromosome. Right here we have different body and eye colors amongst flies, and they've labeled them with colors to kind of show some chromosome maps here, which are the directions of how those genes might be arranged. And now we have what's called polyploidy. So this is the occurrence of 1 or more extra sets of all chromosomes in an organism. A triploid organism is designated 3N, which means that it has 3 complete sets of chromosomes. So you would take 3 times however many sets they have. Many agricultural crops are polyploid. Wheat is 6N, oats is 6N, sugarcane is 8N, polyploid plants often have increased vigor and size. So in plants, this is a great thing. If you're farming and you want to yield more, bigger, better crops, this is actually great. It's not so great in humans or animals, though. Okay, so that's our lesson on Mendelian genetics with gene linkage and what polyploidy is. If you need to go back and review, make sure you do. And make sure you work through your assignments. Ask me, please, if you need any help to help you get through this. Ask me if you need anything at all. And good luck on your assignments and have a great week.